Well, good morning. I'm sorry for this delay. My taxi was arrested. Just if you didn't before coming here. So it was arrested, or that was arrested by a policeman. Well, but anyway, this is our last lecture. Okay. No? Okay, good. Well, and we continue our topic, neurodegenerative diseases, current insight into the pathogenesis, diagnosis, and treatment. Well, and let's start with the second point, new insight into the pathogenesis of Parkinson's disease. Could you remind me, what does it mean? What was the first point? What does it mean? So the first topic, current insight. This is new insight, and what was the current insight? Do you remember that I told you that 30 years ago, neurologists and psychiatrists were absolutely happy with their highly limited knowledge about the pathogenesis of neurodegenerative diseases? Do you remember why? What was the early insight? Yeah,
because he started to transplant stem cells.
Sorry?
Because they tried and have some good experience of transplantating of stem cells.
No, I think that it was because...
No, they did not succeed in the transplantation.
Because they thought that specific neurons is responsible for specific...
Right.
Yes.
Right. Well, so people, they consider that only a very limited population, a certain population of neurons, is responsible for loss of specific functions, like cognitive functions in what disease? The loss of cognitive function in what disease? Alzheimer's disease. And the loss of motor function in Parkinson's disease. Good. Well, but now we're not happy. We're unhappy, but because we received, we have learned a lot about the pathogenesis, and it happens that neurodegenerative diseases are systemic diseases. It means that there is degeneration not only the specific neurons, which play a key role in the certain functions like learning, memory, behavior, and motor function, but many other neurons degenerate in these diseases. Well, so systemic, and not only in the brain, but also in the peripheral nervous system. Well, but still, let's return to. I will tell you in detail the pathogenesis, the mechanism, cellular and molecular mechanisms of the pathogenesis of Parkinson's disease. But believe me that the same scenario of the pathogenesis is the case for Alzheimer's disease and other neurodegenerative diseases. What does it mean? It means that this is a characteristic of the timing characteristic of degeneration of specific neurons. In the case of Parkinson's disease, these are dopaminergic neurons, which are located in the so-called nigrostriatal dopaminergic system of the brain, not particularly, specifically in the substantia nigra. Also, look please, that this is a general characteristic of degeneration of neurons with age. And you can see that the neurons degenerate with a certain rate all over the ontogenesis and in aging people, right? In elder people. Well, but for certain reasons, the rate of the degeneration of dopaminergic neurons is increased. It is increased. Is increased in patients, and the degeneration level, it receives the so-called threshold level. at the age of 60, 65 years, or even later. What does it mean, threshold level? This is a level which corresponds to the appearance of clinical motor symptoms, or in Alzheimer's disease, cognitive abnormality, okay? Well, and you can see that almost 80% of neurons, 70-80% of neurons should be degenerated for the appearance, for the initial appearance of motor symptoms. But the degeneration of this 70% of neurons and the loss of dopamine in the striatum, it happens over 20 up to 30 years period. Up to 30 years period, okay? Well, so this period of neurodegeneration before the appearance of motor symptoms, it is called prodromal stage or preclinical stage. Preclinical stage, okay? And after the appearance of motor symptoms, there is a period for 10, 15 years before death, when motor symptoms dominate, and this period is called clinical stage of Parkinson's disease. Also, I mentioned, I mentioned earlier that there are several mechanisms of the pathogenesis of neurodegeneration. This is death of the neurons, like propagation of endogenous neurotoxins. You remember that? Do you remember what are the endogenous neurotoxins in Alzheimer's disease and Parkinson's disease? There are some protein molecules which are specifically provoke degeneration of either dopaminergic neurons or cholinergic neurons. Yeah, please.
Dopamine.
Sorry?
Dopamine.
No, this is not a toxic factor. Alpha-synuclein. Alpha-synuclein, they say that not exactly alpha-synuclein. Alpha-synuclein is involved in the normal neurotransmission. Huh? Well, so it is absolutely necessary for a normal neurotransmission in the brain. But in pathology, there is a decrease in the activity of degrading enzymes of this protein, peptidases, okay? Well, so, and that is why there is an accumulation of alpha-synuclein, and it's aggregation. Aggregation, well, so small filaments, individual molecules of alpha-synuclein, they aggregate, giving rise to so-called ligands, which are in fact endogenous neurotoxic factors. And what about toxic factors, similar toxic factors in Alzheimer's disease? Do you remember that? Sorry? A little bit louder for everyone. Beta amyloid, exactly. Beta amyloid. Well, so there is another point that there is a beta amyloid which is also aggregated and it becomes endogenous neurotoxin. Okay? Well, and what is the consequence of the consideration of the pathogenesis like this? This is the what are the main conclusions? The first conclusion that actually nowadays we could diagnose Parkinson's disease and Alzheimer's disease only after the appearance and according to the appearance of specific clinical symptoms. Motor disorders in Parkinson's disease and the loss of memory, learning, and adaptive capacity, social adaptive capacity in Alzheimer's disease, okay? Then, what is that? The next step is to study so-called substitution therapy or symptomatic therapy. Well, what does it mean? If there is a loss of dopamine, it means that we should restore the dopamine level, okay? And therefore, dopamine agonists of dopamine receptors are used for this therapy. But the most efficient drug. The most efficient medicine is, what is the most efficient? L-dopa. L-dopa. This is an immediate precursor of dopamine. We cannot use, we can use, in addition to L-dopa, this is the most efficient medicine, we could use agonists of dopamine, I mentioned already, but we cannot use dopamine itself. Why? Exactly. It's not permeable. Why the blood-brain barrier? The dopamine agonist, this is an artificially modified molecule which easily penetrates through the blood-brain barrier. Okay. Good. But what is the result? The final result, the international result of these attempts to help to patient. None of the patient, like patients with Alzheimer's disease, has been cured so far all over the world. Dramatic conclusion. Yeah, dramatic conclusion. Well, what does it mean finally? If this substitution of symptomatic therapy is not effective after the diagnosis, after the clinical diagnosis of Parkinson and Alzheimer's disease, what shall we do? What shall we do to overcome this problem? Sorry? Yeah. Yes, exactly. To develop the earlier diagnosis, long before the appearance of first motor symptoms. For these 20, 30 years, before the appearance of motor symptoms and before the clinical diagnosis of this disease. But if we will succeed to diagnose this disease, for instance, 20 years before the onset of clinical symptoms, what shall we do? Shall we use also substitution therapy with agonists of dopamine or with the L-dopa?
We should somehow stop the degeneration.
Sorry?
We should stop the degeneration.
Exactly. Well, so there is no reason to use to replace dopamine, because at this period, there is no deficiency of dopamine. Well, and therefore we should. What can we do? We could to slow down the degeneration of the neurons by using so-called neuroprotectors. And therefore, if we will succeed to prolong this period of preclinical period of degeneration of the neurons for 20, 30 years, it will result in the appearance of motor symptoms or specific symptoms for only at the age of 90, 100 years. For most people, that is not really very interesting. Yeah? Good. Well, so these people would die for another reason, for another cause, before the appearance of the specific symptoms. This is the main idea. Yeah? Clear? Good. Well, and now what is the current paradigm for the development of preclinical diagnosis of PD, of Parkinson's disease? And I mentioned already, this is the same for Alzheimer's disease. But could you remind me, what is, well, so in Parkinson's disease, the crucial population of the neurons which control motor behavior and which die in Parkinson's disease, these are dopaminergic neurons. And in Alzheimer's disease, what is the specific neuronal population? According to the neurotransmitter, cholinergic neurons. Cholinergic neurons. And what is the location of these neurons in the brain? Hippocampus and cortex. Good. Well, and now I would return again to the main conclusions, which could be done nowadays. Well, so there is a lot of information. about the pathogenesis, about cellular and clinical mechanism of the pathogenesis, about the systemic character of this disease. But what is the most important? What are the crucial points? First, long-term progressive development without manifestation of motor symptoms for 20, 30 years, up to 30 years. Then the emergence of the first symptoms many years after the onset of neurodegeneration at the loss of most of the nigrostriatal dopaminergic neurons and the depletion of compensatory reserves. Well, so in fact, the threshold of the number of dopaminergic neurons in the substantia nigra and the appearance of motor symptoms, it is 50, 60% of dopaminergic neurons. A loss of 50, at least of 50% of dopaminergic neurons, and the loss of about 70% of dopamine, this crucial neurotransmitter, in striatum, in the striatum, okay, in the place of for the projections of the axons of dopaminergic neurons. Good. And the low efficiency of traditional substituted for symptomatic therapy. What is the prospect? I mentioned already, development of preclinical diagnosis and preventive neuroprotective therapy. Well, and this is a very provocative question. Provocative question, because in fact, for the 30 years of the hard work, what are the results of this activity all over the world? More than 1,000 papers were published in good journal, including Nature, Science, etc., etc. Okay, like hundreds of millions of dollars were invested. Also, a lot of foundations which invest to this issue. For instance, in the United States, there is the most important foundation which support the study and the development of technologies in Parkinson's disease, this Michael Fox Foundation. Do you know this name, Michael Fox? Huh? Michael Fox?
You played the main role in the diffusion.
Yeah, Michael Fox is an actor in Hollywood. Hollywood. Well, so he suffers from Parkinson's disease, and therefore he created this foundation. He created this foundation for Parkinson's disease. Well, so this is the most important funding source for the development of technologies in Parkinson's disease. Well, and the main conclusion, the most dramatic, that still there is no technology for preclinical diagnosis of Parkinson's disease. And this is also the case for Alzheimer's disease, okay? You know this picture?
Of course.
Who is an artist, painter? Huh? Who? Okay. Okay, good. Yeah.
So I just listed it. So we say that we have not technology for any diagnosis, but how you figure out that the one set of neurodegeneration is at this age, late 30s or 40 years old? So how to figure out?
How this conclusion was made?
Yes, it's becoming not
Well,
that. It's really
So exactly. This was a mathematical extrapolation of the kind characteristics of the degeneration of the neurons after the appearance of motor symptoms. This type of characteristic was extrapolated by mathematicians for the early stage, before the appearance of motor symptoms. And that's a good question. Well, so and the question arises, is something wrong in our methodology, in general methodology of the development of the treatment of Parkinson, or the diagnosis and treatment of Parkinson's disease? Well, and in fact, there is a mistake, a huge mistake. But the first question, is it possible to diagnose Parkinson's disease at the preclinical stage nowadays? You know already that we can use positive emission tomography for detection of failure of dopaminergic neurons. Please remind me what ligands, radioactive ligands, isotope, can be used for the evaluation of the functional activity of dopaminergic neurons in the strategy. What kind of ligands? Ligands for what natural molecules, functionally important molecules? That's more simple question. What are the most crucial functional molecules in dopaminergic neurons? Sorry? Dopamine certainly, this is the final synthetic program. But I mean the functional proteins, protein molecules.
Tyrosine.
Tyrosine, this is an amino acid. This is an amino acid precursor. It's not a protein. It's not a protein, and tyrosine is widely distributed all over the organs, all over the body, including the brain. But what are the most important functional molecules, specific functional molecules, protein molecules in the dopaminergic neurons? Sorry? Now, I know, but this is also the precursor of dopamine. This is not a protein. This is just a sort of amino acid. Not the
Tyrosine hydroxylase.
tyrosine hydroxylase. What is functional? If it is responsible for what from the functional point of view?
It makes it's in two stages of L-tyrosine and L-dopa. So
It converts
it converts L-tyrosine.
L-tyrosine to L-dopa. Good. Well, the first functional molecule, at least two additional molecules are used for the radioactive labeling for positron emission tomography. That's two molecules additional. Not really tyrosine hydroxylase, it is not labeled, radioactively labeled. What is radioactively labeled? Well, so, no, let's continue. Well, so tyrosine hydroxylase, the first functionally important protein. The next one, the next enzyme, which is involved in the regulation, sorry? ADC, aromatic amino acid decarboxylase. The second enzyme, which converts L-dopa to dopamine, okay? Then, the next functionally important molecule, which is specific only for dopaminergic neurons, not for any other cell and not for any other neuron in the body. There are no other cells except dopaminergic neurons which express this protein. What is a protein? Absolutely specific for dopaminergic neurons. And the only one. Because tyrosine hydroxylase, it is expressed also in all catecholaminagic neurons, not only in dopaminergic, in noradrenergic neurons also. What is? Nobody remember? Dopamine transporter. Good. Dopamine transporter. This is a functional molecule which is expressed only in dopaminergic neurons in the whole body. There is no another cell with dopamine transport. Okay. Well, so and now, what kind of ligands, radioactive labeled ligands, are used for PET scan? What's PET scan examination of the functional activity of dopaminergic neurons. Do you remember? What molecules are labeled with isotope for the study of the functioning of dopaminergic neurons? Well, so precursor of dopamine. How it is called? The immediate precursor. L-dopa. L-dopa. Well, so we use fluorodopa, it is called. And then for dopamine transporter. For dopamine transporter. And then there is, you're right, that there is another important molecule, but which is expressed in dopaminergic neurons and also in the target neurons. Well, so these are dopamine receptors. Okay? Well, so we can use the ligands for dopamine receptors. And please remind me, what does it mean that there is an upregulation between the expression of receptor molecules in the target neurons and autoregulation and the content of dopamine in the intercellular cleft? What does it mean that there is an upregulation between the amount of dopamine in the intercellular cleft and in the level of the expression of receptors in the target neurons? What does it mean, upregulation?
It means that amount of receptors is regulated by amount of dopamine
Exactly. It depends on the amount.
intercellular.
Yes. But what is the correlation exactly? If there is a small amount of dopamine,
It will increase.
there is an increase of the number of receptors in the membrane. Well, so in other words,
To increase probability of catching.
Exactly. Well, so in other words, the low secretion of dopamine is compensated by the increased sensitivity of the target neurons to dopamine via the increase of the number of receptors. Good. Well, but it is written here that that positron emission tomography available for preclinical diagnosis of PD in Parkinson's disease in individual subjects, but not for general preventive examination of healthy population. Why? What's your idea? Yeah?
Well, maybe it is just
It-
impractical because it is an invasive technique.
It is what kind of technique?
I mean, PAT is invasive. We have to do injections, and
You mean that it might be toxic?
yeah, it's maybe it's also expensive.
Good, that's possible. And other ideas? Well, so the first idea that isotopes themselves are toxic, at least to a small extent, and that is true. Well, and therefore I mentioned already that positron emission tomography is not allowed to make it more often than once a year. Yeah, you have an idea?
So I think that generally it's just that's too much selective reporting. So generally we make MRI and it allows us to detect something like conscious or spectrum of diseases. But the technique really would detect specifically the onset of the Parkinson's disease, and I think the reason that people just would not do it massively.
Well, but actually, it is used a combination at the same time of MRI and PET scan. So not individual. That sometimes ago there was a combination for the study, a combination of CT, computer tomography, and PET scan. But now we used a more effective combination of MRI and PET scan. Well, so no, that's not really the case that you say. But what are other reasons? Well, so look, there are two additional technical reasons. First, that PET scan, this is a real, it's not a widely distributed technique. PET scan, it is used in a limited, in a quite limited number of hospitals, of specialized clinics in neurology, psychiatry, etc. On the one hand, and on the other hand, it is necessary to use in the immediate vicinity a very good radiochemical laboratory, because the half-life of these isotopes sometimes it's no more than half an hour, and therefore the people in the radiochemical laboratory should prepare, and it should be used this isotope no for a longer time than a half an hour, very quickly. Well, so and it's very costly also, and also this technique is very expensive. Well, so one examination in for Parkinson's disease in Western countries, it costs no less than $2,500. Well, no, so it is expensive. So we cannot use, I return to the conclusion that this technique we could use for preclinical, theoretically, we could use for preclinical diagnosis of Parkinson's disease, but practically we cannot use it for technical reasons. This is a rare technique, sophisticated technique, and very expensive technique. Okay? Good. Now let's know. Well, and what is actually the idea to develop so-called preclinical diagnosis of Parkinson's disease? I repeat once again that nowadays we are not as happy as it was 30 years ago, and we know that Parkinson's disease is a systemic disease, and in addition to substantia nigra with dopaminergic neurons, there are other brain neurons and the regions of the peripheral nervous system which are degenerated in Parkinson's disease. And in many regions, neurons degenerate before the degeneration of dopaminergic neurons in the substantia nigra. It means that we could imagine the appearance of non-motor symptoms, or premotor symptoms, before the appearance of motor symptoms. What does it mean? For instance, if there is a degeneration of the neurons in the olfactory nucleus, what could we await as clinical symptoms?
Loss of smell.
Exactly. Abnormal smell, and impaired affection. Do you remember I mentioned three types of diseases which are started with the appearance of these olfaction symptoms. Covid first, then Parkinson's disease and Alzheimer's disease. The initial symptoms for all three, and I think that not only for these three diseases, is the mild olfaction, because the toxic factors, they enter the brain via the nasal cranium and via the nerves to the brain, bypassing the blood-brain barrier. And the first target on their way to the brain are the olfactory neurons. Okay? Yeah.
As far as I'm aware, with COVID, olfactory symptoms they are not always present.
No, I'm not really a specialist in infectious disease, but as far as I remember, during this COVID epidemic of COVID, it was said that the first symptoms which we should take into account, this is a mild infection or an impaired infection. Well, nowadays, I'm not sure that there are the different variants of COVID actually, but I'm not sure that this symptom is also as the first symptom is the case in this nowadays COVIDs. I'm not sure, but initially that was really it was generally accepted that the impaired affection is the first symptom. Hmm? Well, and look, but if there is degeneration of many neurons in the brain, and in the peripheral nervous system, or in internal organs, in fact, we could await the appearance of preclinical markers of Parkinson's disease before the appearance of motor symptoms. Well, and this is the case, in fact. And then we could await changes in body fluids. What kind of body fluids do you know?
Blood.
Blood? Yeah, what? Lymph?
Saliva.
Sorry?
Saliva.
Well, so yes, we could say. And other?
Urine.
Urine? Yeah.
Cerebrospinal.
Sorry?
Cerebrospinal.
Cerebrospinal fluid. And the tail fluid, huh? Tail fluid. Tail fluid, okay? Well, but what are the most available body fluids for their examination, for the examination of their composition?
Urine. Plasma.
Plasma, blood,
It should be.
blood, not only plasma, huh? Because we use also the cells, the blood cells, for the study for the expression of specific genes and proteins. Well, blood and urine and tail fluid, huh? And the tail fluid, okay, good. But this is not the case. What is the best body fluid for this, for the developing of or the preclinical diagnosis?
Cerebrospinal.
Cerebrospinal fluid. But it is not available. In fact, it's not available, at least in Russia, huh? Because in Russia, even neurosurgeons, they do not agree to take cerebrospinal fluid, or because there are very rare, but there are some side effects and some complications, infections, etc. After taking the cerebrospinal fluid. But this is not the case for American people. American people, they easily do it. And then patients, they're very enthusiastic in giving this material to physicians because this is doing for the development, for the facilitating of the state, of the life of real patients. This is a moral stimulator for American people. That is quite different from our people. Well, for instance, we're developing this preclinical diagnosis in my laboratory, and all my associates, they gave their blood and their tail fluid as a control, right? but not myself. Never give. Well, and now let's please, what is the current and upgraded developing technologies for early diagnosis of Parkinson's disease by searching for markers and body fluids? Well, so what are the methodologies, in fact? Well, and for a long time, there was a search for biomarkers in untreated PD patients at the early clinical stage. Well, so the patients, just after the appearance of motor symptoms and the diagnosis of Parkinson's disease, they are examined in terms of premotor symptoms. And what are the premotor symptoms? The first one is malinfection, okay? And then this is the impaired sleep, huh? But the specific impaired sleep, when people are very aggressive during their sleep. Huh? Well, so and changes in body fluids. But look, please, at this schematic representation. Well, so we could imagine, well, so according to our hypothesis, there is a small number of specific preclinical markers in body, in the blood, for instance, or in any other body fluid, very specific. But after as the disease develops, there is an appearance of many other markers, particularly of markers which appear at the clinical stage. And when we searching these markers at the clinical stage, we detect not only the marker of preclinical stage, but also the markers of the clinical stage. And we cannot differentiate them when we're getting this blood with both types of markers. And therefore, it is written here that what is a drawback? No guarantee that all markers found at the clinical stage are characteristic of preclinical stage. And also there is another drawback, that all markers detected so far in patients are non-specific. What does it mean? That if we detect a change in the composition of the blood at Parkinson's disease, we could find the same change in a number of other diseases, neurological and mental diseases. Therefore, all the markers are not specific, but we should try to find out and to create a panel of dozens of hundreds of these markers to make this panel more specific for the diagnosis of Parkinson's disease and any other diseases. Well, so in this case, I think we're coming to the idea of Confucius. It's very difficult to find out a black cat clinical biomarkers in a dark room in the patient at the clinical stage, especially if there is no cat, if there is almost no overlap of blood markers at the clinical and preclinical stage. Well, and the methodology so far, the methodology for the development of this preclinical diagnosis was based on the search of biomarkers which are characteristic of both patients at the clinical stage and the experimental model of preclinical stage, because we cannot take blood or any body fluids in patients at the preclinical stage, because we cannot diagnose this disease at this stage. But we could make a model, experimental animal model of preclinical stage, and we could search biomarkers in the body fluids in animals. Well, and also the model of clinical stage. Well, so the overlap should be a set of biomarkers of the preclinical stage. Difficult to understand? Well, I'll repeat once again. Well, so look, please. We have to detect only the markers of preclinical stage of Parkinson's disease. But we cannot do it in patients because there is no diagnosis of the disease at this stage. And therefore, we could find out the markers of Parkinson's disease at the clinical stage, but at this stage, there are not only markers of preclinical stage, but also additional markers of clinical stage. And therefore, to select from this composition, to select only these markers we should test markers in patients at the clinical stage, then the markers in the experimental model, in animal models of preclinical stage, and the overlap of these markers should correspond to markers of the preclinical stage. Yeah?
About markers at all in this moment, where are they found in blood?
Theoretically, it could be found. It could be found in any body fluid, but the most efficiently the cerebrospinal fluid, which is not available in fact. But the second one is the blood. Okay. Well, and
I'm sorry, can you please
Yeah,
specify?
yeah.
I don't quite understand why should we find exactly where it's left, the clinical stage and the preclinical stage, when we are more interested in the preclinical stage.
yes, sure.
But why do then on the scheme we see it's a combination, like it's a correlation between the two stages, or did I not understand correctly?
Yeah.
The specific markers, they have clinical stages, factor in clinical stages.
You know, but we proceed from the idea that we cannot, that there is no any model, adequate model of any disease. Well, so if we are doing a model, experimental model of Parkinson's disease, we could follow only to the individual signs, individual features of this disease. Well, so in this particular case, the model, they reproduce only the degeneration of dopaminergic neurons, but not of all the neurons, systemically. You understand? Therefore, there is a limit of the use of these markers of preclinical stage. Well, sorry. But nowadays, there is the most efficient way to develop preclinical diagnosis directly in patients, not in animals, but in patients. Well, so we could search, we could make a diagnosis of preclinical stage of Parkinson's disease nowadays according to preclinical premotor symptoms. And therefore, we could take, in these patients, we could take blood for searching biomarkers. But still, I repeat once again that that this methodology for the search of markers in body fluids, this will be always non-specific, because we finally will be able to say that there is a risk of the development of Parkinson's disease with the 90% of the probability, for instance, or 80%, but we will not be able to say for 100% of the probability. Well, and then another point, development and specification of experimental models of Parkinson's disease at the preclinical stage. When we are comparing the spectrum of the blood markers, for instance, in patients and in animal models, we should develop more or less adequate models of Parkinson's disease. And we have done it. And not only of Parkinson's disease at the clinical stage, but also at the preclinical stage. It means that we should reproduce in animals when we are modeling the preclinical stage, we should reproduce in animals, like in patients, the loss of dopamine in the striatum for 70–80%, less than 70–80%, before the threshold level, and a loss less than 50–60% of dopaminergic neurons. Okay. Well, and we have developed actually in our laboratory these two models, preclinical and early clinical stage of Parkinson's disease. The preclinical stage, there is a loss of less than 70% of dopamine and dopamine actions in the striatum, a loss of less than 50% dopaminergic cell bodies in the substantia nigra, and there should not be motor disorders in these animals. Okay? And at the same time, there should be systemic loss of central and peripheral neurons and bad visceral functions. What does it mean, visceral functions? For instance, sympathetic neurons, which use norepinephrine, like dopamine, for the regulation of internal organs, like cardiovascular system. They also degenerate in Parkinson's disease. At the preclinical stage, in patients, only 50% of patients at the preclinical stage, they have an impaired innervation, sympathetic innervation of the cardiovascular system. Well, the requirements for modeling Parkinson's disease at the early clinical stage. This is a loss of more than 70 or more than 70-80% of dopamine. The loss of more than 50, 60 or even more percent of dopaminergic neurons in the substantial network, and the appearance of motor disorders, and systemic loss of central and peripheral neurons and impaired visceral functions. Well, so we have developed these models by using specific neurotoxin of dopaminergic neurons. Well, so in fact, we use the precursor, pretoxin. This is a molecular precursor of toxin of dopaminergic neurons, NPTP. But after the injection, systemic injection to the animal, NPTP is converted in glial cells into the effective toxin of dopamine, specific toxin of dopaminergic neurons, NPP. Okay, and this toxin provokes the degeneration of nigrous striatal of dopaminergic neurons. The cell bodies are located in the substantia nigra, and the axons are projected into the striatum. And this is a region, specific region, for the control of motor behavior. Okay? Well, the people they began to use NPTP after an accident in the United States, because it happens that in '86 there was a publication in the United States about a group of a number of young people, druggers, which used heroin, heroin or German.
Heroin.
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 Heroin. Well, so and the basis of heroin, that was NPTP, and that is why it is used actually widely used for the development of or the model of Parkinson's disease. Well, so in these people, these young people, they demonstrated motor symptoms like Parkinsonian young people. Well, and certainly when we're modeling something in animals, we should be, it is necessary to be sure that we reproduce correctly at least some features of the pathogenesis in humans. Okay, and therefore we compared the gene expression of the crucial proteins which are involved in the development of Parkinson's disease. This is alpha-synuclein, we have discussed already, and these are proteins which are involved in dopamine neurotransmission. Well, so we evaluated the expression of these genes in different experimental models of Parkinson's disease, in preclinical model and the clinical model. And we compared the expression of these same genes in patients with Parkinson's disease to be sure that we correctly reproduce these diseases. Look, please, there is a full overlap between the expression of genes in patients and the expression of genes in our animal model of clinical stage. Okay, it is naturally that these characteristics are different in the preclinical mice. Huh? Well, but the most important that we correctly reproduce, at least according to the expression of the genes of these proteins, the pathogenesis of Parkinson's disease. Well, and then we tested 13 markers in the blood in patients with Parkinson's disease at the early clinical stage, in non-treated patients, which is very important, huh? And in preclinical mice, clinical mice, and TB patients at the early clinical stage. Well, so we compared, and there is an overlap only between 13, only in three markers. L-DOPA, this is a precursor of dopamine, an immediate precursor of dopamine. DAPAC, what does it mean, DAPAC? This is the product of degradation of dopamine, enzymatic degradation of dopamine. Okay, DAPAC. Okay, and also in the expression of gene for dopamine in lymphocytes. Well, so when saying that we detect markers in the blood, I should emphasize that there are two parts of the blood: the plasma, and we test the chemical composition of plasma and its change. And also there are cells. There are cells which modify their functional activity in Parkinson's disease, and these are lymphocytes. It is curiously, I have learned it myself. I think 20 years ago, and it was a great surprise for me, that lymphocytes express the same phenotype as dopaminergic neurons. Good? Well, so chemically, they're identical to dopaminergic neurons. They express both enzymes, so dopamine synthesis, tyrosine hydroxylase and decarboxylase. They express dopamine transporter, and they express the receptors for dopamine, including outer receptors for dopamine. And therefore we studied not only the composition of the chemical composition of the plasma, but also the gene expression in lymphocytes. And we found between three genes which were tested, we found a change in animals, in animal models and in patients, only receptors for dopamine type 3. Okay? Well, so, and finally it happened that I told you that between 13 markers, we found an overlap only of 3 markers. It means only 23% of markers which were found in the blood of patients can be used for preclinical diagnosis of Parkinson's disease. Understand? Good? Well, and then we tried, I think for the first time in literature, we tried to find out the changes in tear fluid in PD patients and in Parkinson's animals. Why we could equate the change in the tear fluid in the tear fluid? Huh? Do you have an idea? Well, sure. When studying the composition of the tear fluid, our hypothesis is that should be a change in the composition of tear fluid, and it would be nice if there is a change in the composition of in the content of catecholamines or their metabolites, etc. Why we await that there should be changes and there is a content, there is a certain amount of catecholamines and of their metabolites. What do you think? Because look, I emphasize several times that Parkinson's disease is related not only to the degeneration of dopaminergic neurons, in the nitrostriatal system and the brain, but also it extends to peripheral sympathetic neurons. And the eye is innervated by sympathetic neurons which produce catecholamines. And therefore we could equate then in the tear fluid we could find the changes in the composite in the amount of catecholamines like norepinephrine, epinephrine, dopamine, and the precursors, the product of their degradation, and also in some specific proteins like alpha 2 microglobulin activity. And we tested this 1, 2, 3, 4, 5, 6 potential markers, but we found the overlap only of 2 of them. This is norepinephrine, and this is alpha microglobulin activity. Well, so according to our idea, these 2 markers, or 70% of markers in the tear fluid, could be used for preclinical diagnosis of Parkinson's disease. Good. But I repeat once again that certainly if we are looking, if we're searching for biomarkers in patients at the clinical stage and even at the animals, it's not really sure that we will find the specific or at least semi-specific markers of preclinical stage. And there is another methodology which is not widely, it is used in a small number of laboratories, including our laboratory. According to this methodology, first we're trying to create a risk group of people, of patients, at risk for the developing of Parkinson's disease at the preclinical stage. Well, so between healthy people, we're trying to find out to select elderly subjects without motor symptoms, without diagnosis yet Parkinson's disease, but with premotor symptoms like impaired sleep, I mentioned, olfaction, and also it's very important the impaired peristalsis. Yeah, sure.
Yes, Parkinson's disease have some sort of like genetic basis.
Yes, certainly.
Can we then why don't we test people who have like genetic predisposition? I mean, if they do like a genetic test and they know that they have a predisposition, for example, we can say that to them and like
Okay.
ask them.
Okay, it's clear. Well, probably I mentioned once or 2 times, but it doesn't matter. I will repeat once again. Well, so for many years, the people they selected, they determined a number of so-called risk genes for the developing of Parkinson's disease and Alzheimer's disease. And finally, these patients with Parkinson's disease and Alzheimer's disease, they are included in 2 groups. In one group, patients are included with so-called this is called monogenic group. And another one, polygenic. What does it mean? That in monogenic group, there is the whole set of the genes for risk for the developing of Parkinson's disease or Alzheimer's disease. And therefore, the disease would be developed with certainty, without exception. And another group, this is polygenic group, where there is an expression of a number of risk genes, but the expression strongly depends on so-called epigenetic factors. And therefore, this could be expressed only under a certain combination of epigenetic factors. For instance, on the appearance on the coming into the brain of exogenous toxic factors like iron, like pesticides, etc., etc. Well, so the first group is called monogenic group, and another one polygenic. What is the proportion between them? The monogenic corresponds to there are 5% of all the patients which suffer with monogenic disease, and 90 Parkinson's disease. Alzheimer's disease at a little bit more, 10%. And the polygenic, there are 95% with polygenic or sporadic disease. Well, so we're studying and talking only about these polygenic patients. Okay. Well, and the patients with the monogenic disease, so the motor symptoms appear very early in their life, in age no later than in 30, at age 30. Huh? Okay. Well, so and they die very early, and the physician they can do nothing after that. Well, and so certainly this gene expression is studied, and it is used as markers, not only blood, but these genes are studied mainly in patient in real humans, in the lymphocytes. As I told you, that the lymphocytes express the same genes, the risk genes, and therefore we can study the risk genes for the development of Parkinson's disease or Alzheimer's disease by studying that's not well. Well, but let's continue. Well, so first we create a risk group. A risk group with patients with a risk for developing of Parkinson's disease at the preclinical stage. Then we search for blood markers, chemical composition, gene expression in lymphocytes, and amino acids, monoamines, etc., etc., in the blood or in the tear fluid and in the tear fluid. And there we select a panel of the candidates for the preclinical diagnosis of Parkinson's disease. And then we test whether we are right with the diagnosis of Parkinson's disease at the preclinical stage by using in a small group or a risk group, in a small risk group, by using positron emission tomography. Well, so for instance, in our laboratory, we selected during 2 recent years, we selected 26 patients at risk of developing Parkinson's disease at the preclinical stage. 26. Well, so we detected biomarkers in the blood. In this area. And then we made positron emission tomography for 17 persons. 17. After they did not agree to do it, that was the only reason. Between 17 persons, we found 14 with the impaired functional activity of the non-striatal dopaminergic neurons. And what is the final and the most important arguments in the efficiency of this methodology? That is that in 2 years after the neurological examination, 3 patients between 20 showed motor symptoms. Well, so it means that in these 3 patients, we made a preclinical diagnosis 2 or 3 years before the appearance of motor symptoms. And this is an absolute evidence, absolutely convincing evidence of the efficiency of this methodology, and where used it in Moscow and I think in 4 or 5 other teams in other countries, particularly in Denmark. They are very good in this field. Well, so then the second question. I always say to you that this technology for diagnosis or preclinical diagnosis by using blood markers, body fluid biomarkers, will be always non-specific or semi-specific. And therefore the next question could be, but whether there is a possibility to develop a really specific preclinical diagnosis of Parkinson's disease or Alzheimer's disease. Well, and this is a question. And we turned our attention to internal medicine, and it happened that in internal medicine, the preclinical diagnosis of chronic diseases was developed, specific diagnosis, was developed decades ago, and it is widely and very successfully used so far. And I should attract your attention to the point that the scenario of the development of chronic diseases, of brain diseases and of internal organ diseases, is the same. Huh? So these diseases are developed for years or for decades without the manifestation of the clinical symptoms. But after the manifestation of the clinical symptoms, there is nothing to treat. The regulatory systems are degraded, and it's almost impossible to principally to improve the situation. What kind of, well, and for instance, you know that I will give you just a few examples of these chronic diseases of internal organs in internal medicine. There's a diabetes. Diabetes is developed for many, many years before the appearance of clinical symptoms. And for the diagnosis at the preclinical stage, people use so-called provocative tests. Well, so you could imagine that if you inject to a healthy one into a person at the preclinical stage the same amount of glucose into the blood, and then you would monitor the concentration of this glucose. Well, so, and glucose, it should be metabolized in the body. And what do you think the rate of this metabolizing would be higher in patients at the preclinical stage or in healthy people? In health, certainly, because there is a most efficient insulin, the enzyme for degradation of glucose. And therefore, in internal medicine, the so-called provocative tests or challenge tests are used for decades very successfully. And this is the only way to make preclinical diagnosis. And what is the definition? The provocative test or challenge test is any procedure in which a suspected pathophysiological abnormality is deliberated and irreversibly strengthened, causing a short-term characteristic reaction or symptoms of the disease. Well, unfortunately, almost have no time. But let's tell you that we have developed in my laboratory, but it has never been used and developed in neurology and psychiatry. Well, and I discussed this point. That was very surprising for me. And I discussed this point at many international congresses with the most outstanding scientists in neurology. Why people they did not use this provocative test in neurology? for any chronic disease, not only for Parkinson's disease and Alzheimer. And there was no response. Huh? And the only one I did was that the medicine historically is developed from two big principal stones, let's say stones, cores. That's neurology and internal medicine. And there is no always overlap between these two branches, historically developed branches of medicine. If there is a congress on cardiovascular research, for instance, in cardiovascular system, there are all the representative of the internal medicine. There are pulmonologists, there are endocrinologists, there are nephrologists, but there are no neurologists, almost no neurologists. But on the other hand, if there is a congress on neurology and mental diseases, there are neurologists, there are neurosurgeons, there are neuropharmacologists, there are no specialists in the internal medicine. And therefore, the only one reason for this surprising shift in the development of provocative tests, to my mind, this is the absence of the overlapping of these two medical societies. Well, sure, unfortunately, we have no time, but you could believe me that we can develop in our laboratory for the first time in neurology and in psychiatry the provocative test for Parkinson's disease. What was that? That we have used as a provocative agent. We have used an inhibitor of the first enzyme of dopamine synthesis. I think methyl P-tyrosine. And this is a reversible, non-metabolizable inhibitor. Well, so after its action, and we could receive a dose-dependent effect. But after the use of this inhibitor, it is there is a full clearance of this inhibitor from the organism. Well, and so we have demonstrated, in fact, we have developed an experimental model that at a certain level of the preclinical diagnosis, if we use alpha MBT, we could deplete dopamine until the threshold level, thereby provoking the appearance of motor behavior, of motor disorders. But this is not the case in the control animals with 100% of dopamine in the strain. Well, and really the last slide. Well, so in neurologists, they're very conservative, much more conservative than specialists in internal medicine. And therefore, nowadays, we should make good clinical trials just to prove, just to give evidence that this is not toxic for patients and for people. But there are so many arguments against the toxicity of this inhibitor. Well, so it is widely used in healthy subjects to study with positron emission tomography the molecular mechanism of dopamine neurotransmission. It is widely used for the clinical use for the treatment of so-called pheochromocytoma, with the excessive, with overexpression of norepinephrine, and for the treatment of many other other diseases, with no manifestation of toxic effects. Well, so this is a general scheme for the development of the early preclinical stage of Parkinson's disease. Well, so this is the search for biomarkers, the use of provocative tests, and the testing, finally, in the risk group, the dysfunction, the functional insufficiency of the nitrous dopaminergic system with positron emission tomography. Well, and let's come to the conclusions. Dozens of people suffer from neurodegenerative diseases, and this, the number of the incidence increases rapidly. None of patients with neurodegeneration has been cured. Neurodegenerative diseases are developed over decades without specific symptoms. Dramatic symptoms first appear at the death of majority neurons. Neurodegeneration is not limited to the brain, but extends to the periphery. One of the global challenges in neuroscience is developing preclinical diagnosis. And the specific preclinical diagnosis may be developed by using provocative tests. And preclinical diagnosis of neurodegeneration would promote the development of preventive treatment, but for stopping or at least slowing down neuronal disease. Well, so this is our last lecture. I remind you a list of the exam list for your questions. You could prepare even in advance if desirable for your exam any question. Well, and the exam will be at 28th of March from 9:30. Well, and thank you very much for your attention, for your time, and for assisting my lectures. And what is the most important today? Specifically today, what is the most important?
Questions?
Sorry?
Question?
You have no good fantasy. What is the most important?
Development of preclinical diagnosis for Parkinson's.
Poor guys. This is the most important. Poor guys. You're not happy with your friends, boyfriends. Sorry?
Tomorrow.
Yes, tomorrow, but we will not meet each other tomorrow. And you should express your greetings today, I think. Because you are not happy with your guys. In my generations, girls were more happy. Believe me. Okay, thank you very much for your attention. We have a break, and after that we'll get the full presentation. Yeah, see you.